All right. Welcome to those of you who are new to Malaysian Research Insider, and to those of you who have joined us previously, welcome back. I'm Ayapa. I'm doing a PhD in molecular medicine at University of Science Malaysia, and I'll be your host for today's Research Insider session. This science chat is co-organized by MBIOS, 100 Scientists of Malaysia, along with our media partner, Science Media Center Malaysia. Right, so welcome our scientist for today, Sean Wen, our fellow Malaysian pursuing his PhD in medical sciences at the University of Oxford. Okay, I will allow him to introduce himself further and what he is working on. Over to you, Sean.
Okay, so I think it's best for me to do the introduction through the slides. So is
Yeah.
this how I share my slides? Okay. Sorry, so I guess I can move forward from here. So thank you so much for taking part in the poll. Today I'm going to broadly bring you through my research experience since I graduated with my bachelor's degree. And then I'm going to talk about a little bit of the work that I'm doing currently at the University of Oxford. Essentially, I'm trying to review cancer heterogeneity by looking beyond genes. And soon you'll find out what do I mean by that. So today we'll go through 4 themes in total. The first theme will be about inheritance, and then we'll talk about what fidelity means in the context of cancer, and followed by myths. And lastly, I'll just speak very shortly on how the corona pandemic has affected cancer research. So I joined the International Medical University. Specifically, I went through the Bachelor of Science Biomedical Science program. So essentially, it was really a lab-based degree where we kind of cover a lot of different modules. So it's like a buffet. You know, we covered anything from hematology, microbiology, the immune system, genetics, histopathology. This was one of my this was one of my favorite module. And then we also covered briefly on the different human systems. And I think largely I went to do this program because if you were like me, who was born in the early 1990s, you basically grew up watching the CSI series. So I was very much inspired by the forensic science and how they use the different laboratory techniques to catch the bad guys. So after I graduated from IMU, I joined Cancer Research Malaysia, where I studied inherited mutations in breast and ovarian cancer patients. And so that brings us to our first theme for today, which is inheritance. So how common cancer is, I guess sometimes it also boils down to a matter of perspective. So for me, you can see this is me in the middle, and I don't personally know anyone in my family or my friends who have cancer, but I do know some friends of mine who do have cancer. So I know somebody who has a developed lung cancer when she was more than 60 years old. And just this year, I found out that a friend of mine had colorectal cancer, diagnosed at the age of 60 plus. And I think just 2 months ago, I heard that a friend of mine developed ovarian cancer around the age of 30. So another way to formally look into the cancer frequency in one's family is through looking at pedigrees or family trees, as drawn over here. So let's say I'm interested in this case over here, the point of reference. So this is the index case. So in this index case over here, you can see that she is a female. And so she's married, and this is a husband, which is male, which is in square. So you can see that her mother was diagnosed with ovarian cancer at the age of 49. And then her aunt was diagnosed with breast cancer at the age of 52. And so if you move up the family tree one more generation, you would see that her grandmother was diagnosed with ovarian cancer at the age of 45. So in this particular case, the cause of the cancer can be traced back to an inherited genetic mutation in the BRCA1 gene. And when the index case, though she has not developed any cancer, but she went for genetic testing, and she found that she was also a carrier of BRCA1. So this family tree actually belongs to Angelina Jolie. So when we look into the prevalence of the BRCA1 and 2 genes in Malaysian ovarian cancer patients, we found that among 218 patients, we found that 1 in 10 turned out to be BRCA1 and or 2 carriers. When we look into the prevalence of BRCA1 or 2 gene mutations in Malaysian high-risk breast cancer patients, what we found amongst the 100 cases and high risk, so high risk here means that these patients were typically diagnosed at a young age of diagnosis, and they have a strong family of history. So when we look into these high-risk breast cancer patients, we found that similar to ovarian cancer patients, the prevalence of BRCA1 and or 2 carriers is 1 in 10, so 10%. When we look into a larger cohort of breast cancer patients, in this case, we look specifically into 2,500 Malaysian breast cancer patients. We found the prevalence of the BRCA1 and 2 gene to be 1 in 20, so that's 5%. We also assisted in a project where we look into breast cancer patients of European descent. So this was a larger study, 2 times larger than the Malaysian cohort at that time. So this cohort included 5,000 European breast cancer patients. So you can see that in this particular cohort, we found that we found the frequency of BRCA1 and 2 carriers to be 1 in 50. So much lower compared to their Asian counterpart. So in parallel, I also did my master's with University Malaya, and so for my master's project, I look into a different kind of gene mutation. So this gene is called ApoBac3B, and we found out that this genetic mutation in this gene is pretty common in the Malaysian population. So when we look into 1,500 healthy individuals, what we found was that half of the women carries a genetic mutation in ApoBac3B. And what's interesting is that the percentage of ApoBac3B carriers across the ethnicities are slightly different. So you will see that they're more common in the Malays, followed by the Chinese, and then lastly, the Indians. And so when we compare the frequency of the ApoBac3B gene deletion in the breast cancer cohort, we found that 1 in 6 of these women carries the genetic mutation. But what's interesting is that when we look into the tumor profiles of these patients, we found that tumors arising from breast cancer patients with this ApoBac3B deletion tend to on average have a higher abundance of immune cells surrounding the tumor compared to tumors that arise from breast cancer patients without this genetic mutation. So after that, I then briefly returned to IMU, and that brings us to our next theme for today, which is Fidelity. So in this slide, I'm just going to show you what is the consequence of having an inherited mutation compared to a mutation that you acquire later in your life. So let's say we have a time scale over here. So on the left-hand side is where life starts. So this is where you're born, and then we start, living the rat race, and then we rush our way throughout life to get to old age. So in the beginning, cells would divide as they do, and then perhaps somewhere along the way, you may acquire a genetic mutation. it could be due to it could be due to error during the DNA replication process, or it could be due to potentially environmental insults. But whatever is it, sometimes unluckily we do acquire an important mutation along our lives. But of course, one mutation is potentially not enough to give rise to cancer. So, but nevertheless, that one mutation that is acquired in the beginning will be sort of inherited down the road to all the cells that arises from that cell. And then maybe unluckily, you may get a second mutation somewhere along your life. And then the cell continues to divide uneventfully, but potentially above a certain threshold or above a certain number of mutations, that cells becomes really unstable, and then it starts to give rise to a clone of cells in which what we call a mass of tumor. So that is why typically in the absence of inherited mutations, or potentially in the absence of any obvious environmental factors, tumor tends to arise in particular during old age. But the scenario is a bit different when we talk about a person who has inherited a genetic mutation from the start, potentially from their parents. So in this case, every cell of their body would have, let's say, the first mutation because it's inherited. So the cells may go on to divide uneventfully at first, but eventually potentially a second mutation will arise. And then once the cell accumulated enough genetic mutations, it would then start to divide uncontrollably and start to give rise to that mass of tumor. So you can see how the impact of the first mutation in terms of on the timing in which a person acquires the tumor. So I haven't really explained to you what are the roles of the BRCA1 and BRCA2 gene. So in this slide, I'm going to explain to you what they do. So essentially you have the DNA over here and you have the cell. And then so the cell would divide as usual to replace old and dying cells. But sometimes during the replication process, the cell doesn't faithfully copy the entire DNA exactly as it is to the next cell. So this is what we meant by fidelity. Fidelity means that it's the ability of a cell to copy its DNA 100% correctly to the next cell. So sometimes you get a genetic mutation during that process. So over here, I'm showing you an example of a double-stranded break where you get a portion of the genome that does not get copied. But fear not, if you have DNA repair genes that are working well, this damage over here can be repaired. And so once it's repaired, then the cell can go on to divide as usual. But sometimes we get mutations in the DNA repair gene, and sometimes what happens is that the DNA damage does not get repaired. And so when the damage is not repaired, what you get is the beginning of the accumulation of more and more mutations down the road. So this brings me to my work in IMU. So what I wanted to do over here was that I really asked myself the question of, can I do more for BRCA1 and 2 mutation carriers? Well, mainly because is that it is bad enough that you inherited something that you did not ask for, and moreover, that particular gene increases your risk of lifetime cancer to breast and ovarian cancer. So this was purely a computational exercise. So for this study, I asked myself, are there any differences in the features of tumors arising from the BRCA1 and 2 compared to tumors with functional BRCA1 and 2, and whether we can leverage those features for targeted therapy? So in this exercise, I downloaded 500 whole genome sequence breast cancer and another 1,000 whole exome sequence breast cancer samples. And so when I look into the number of mutations that arise from this tumor, quite naturally and quite expectedly, I found BRCA1 and 2 tumors to have higher number of mutations compared to, on average, compared to tumors that has functional BRCA1 and 2. And so whenever a cell acquire a genetic mutation, it also tends to express a new proteins, and these new proteins get in turn expressed on the surface of the cell. So when I look into the BRCA1 and 2 tumors, quite naturally, I also found that the tumors arising from this from tumors that has this genetic lesion tend to have more of these new proteins. And so these new proteins are typically recognized by the immune system, in particular, for example, natural killer cells. And what I found was that the tumors arising from BRCA1 carriers tend to have a higher abundance of immune cells. And this could mean that the BRCA1 can be a potential candidate for immunotherapy. But in contrast to tumors arising with functional BRCA1 and 2, they on average have less immune cells surrounding the tumors, mainly because there are less novel proteins on the surface to instigate or stimulate the immune response. But what was really ironic was that despite BRCA2 having quite a lot of mutations and a lot of novel proteins on the surface, it did not quite stimulate the immune response. So this was the differences that I was quite interested to investigate further. But unfortunately, at that time, my time was up, so I had to move on. So I guess the differences in the immune response between BRCA1 and 2, that question, I think I would have to probably wait for another day to answer that. So after being in IMU briefly, I then joined IQVIA as a data analyst. So it's basically in a non-research setting at that time. And then after that, that very same year, I then proceeded to the University of Oxford to pursue my PhD. So that brings us to the third theme for today, which is myths. So I think we're all very familiar here that genes essentially codes for a sequence of nucleotides, and that those sequence of nucleotides essentially gets transcribed and translated into proteins, or I'll call them gene products over here. And proteins are really the workhorse of our cells because they really do all the heavy lifting. So once upon a time, people look at genes as something of like a through the lens of a one genes, one protein hypothesis. So a long time ago, people used to think that one gene will only give rise to one protein product. But we now know that that's not the case for more than 95% of the genes. So in truth, one gene can give rise to multiple gene products. And so for simplicity, I'm only going to show an example where a gene gives rise to two gene products. So in this case, we're going to call them gene product A and gene product B. And so gene product A can give rise to gene product to a protein that has function A, and gene product B will give rise to a protein B that has protein function B. And so the analogy goes like this. You can see that at the bottom over here, you see two pictures of apples. So these two apples represents the same gene because, you know, both are apples, isn't it, right? But if you look more closely, you can see that the puzzle pieces that make up these two apples, they are similar, but they are different at the same time. So this is exactly what I mean by multiple gene product coming out from the same gene. And so why does it matter? So why does multiple gene product from the same gene matters? Why should we bother to look at that kind of resolution? So occasionally, different gene products that arise from the same gene can have opposing functions. So for example, the BCL gene, one form of its gene product promotes cell death, while the other suppresses cell death. Another example is the PKN gene, which is where one form is typically expressed in the healthy state, and then another form is typically expressed in cancer. The VEGF gene, one form suppresses the blood vessels formation in cancer, but the other form promotes blood vessel formation in cancer. And when it comes to the androgen receptor gene, one form confers sensitivity to therapy, but on the other hand, the other form confers resistance to cancer therapy. So my job at Oxford at the moment is really trying to understand what is the landscape of the multiple gene products at a single cell level compared to whole tumor analysis. And in the next few slides, I'm going to show you why single cell approaches may be advantageous in certain aspects compared to whole tumor. So you can see over here on the left-hand side is the whole tumor in its entirety. And so when you look at the mass of the tumor, the entire mass of the tumor, most of the time it's not going to be a heterogeneous mass of tumor. The reality is that tumors are going to be made up of many smaller components, such as different cell types, as you can see over here. So in terms of the readout, you can see that if you study at the whole tumor level, your signals are typically mixed altogether. But if you look at the single cell resolution, you can then see that the signals can be nicely separated out, giving us a more cleaner analysis. Sometimes you want to study single cells at the single cell level because sometimes different cell populations in the same tumor may harbor different combinations of genetic mutations, and sometimes that can lead to a very different sort of cell profile, mainly because of the different kind of mutation combination that each cell population carries. And the tumor itself, when you look at the tumor mass itself, is normally not just pure tumor. You can also have other kind of cell types that are intertwined with the tumor cells. So majority of the cells would probably be tumor cells. And then sometimes you find a subset of cell population that belongs to the microenvironment. So we call these cells the stroma cells. And then you also can find immune cells as well, sometimes at the periphery of the tumor. And so, as you know, the tumor, so you can see that there are different cell types that exist in the tumor sample itself. And mainly because the tumor don't exist in isolation, the tumors are essentially having a very intimate interaction with the microenvironment as well as the immune system. And single cell approaches can help you to delineate the different cell types and to study their biology separately. Another aspect that is probably important, in particular for my research, is that there are cell types that are going to be sensitive to therapy, for example, but sometimes there are going to be rare cells that are sitting between the tumor itself that are going to confer resistance to therapy. And it is these rare cells, and these cells are typically stem cells in the case of blood cancer, that are going to confer resistance to therapy. And these are really the kind of cells that you want to study in depth. But in order to study in depth, you would have to isolate them in the first place. So single cell approaches will allow me to study rare cell types in this case. So if that's not clear enough, here is my final analogy for whole tumor versus single cell approach. So on the left-hand side, you have a really nice dish, all right, the famous egg fried rice, correct? So if you look down at your plate and you saw the egg fried rice, it's probably you probably would take some time to see what are the different components that make up this egg fried rice. But if you go with a single cell approach over here, you will then start to see that, it becomes extremely clear to me what are the components of the tumor. So in this case, what makes up the egg fried rice is are carrots, peas, corn. You have your garlic, ginger and onion. And then of course you have egg, and then you have rice. So after talking so much about multiple gene products and single cell, I'm going to show you an application of studying multiple gene products at a single cell resolution in this slide. So for simplicity, I'm going to show you one example. So this gene PKM, it expresses two forms. The first is A and B. And so what we did in this study was that we studied the expression of these gene products when the pluripotent stem cells are induced to differentiate into newer progenitor cells and motor neuron cells. So you can see over here, this is essentially just an expression signature heat map. So yellow means low expression, blue means high expression. So you can see for the stem cells over here, the induced pluripotent stem cells over here, they primarily express the gene product A, but not gene product B at all. When you look into the newer progenitor cell population, you will see that about 50% of the cells express the gene product A, and another 50% express the gene product B. But when we look into the motor neuron population, you will see that a small population expresses gene product A, but a majority of the motor neuron cells express gene product B. So I think this slide nicely summarizes the application of studying multiple gene products at a single cell level. So, so I think the first 2 years of my PhD has very much been focused on testing out the feasibility of studying multiple gene products at a single cell resolution, because I think nobody knew before this if it was even possible to begin with, but now we know that it is. And also my first 2 years was very much focused on setting up analytical pipeline to handle this kind of data. So I apologize if I'm not going to speak a lot, or I didn't speak a lot on blood cancer today, but if we were to have a conversation again 2 years down the road, I think I can probably give you a nicer picture on the study of multiple gene product at single cell resolution in blood cancers, and what does that mean for therapy and prognosis. So I guess this year is the year of the pandemic, isn't it, right? Because it's impossible to talk about 2020 without mentioning the coronavirus. And I think as researchers, we ought to be very, very alert and mindful about the impact of the pandemic on our respective area of research. So I'm just going to very, very briefly speak about what the impact of the coronavirus has been for cancer management. So with a very good friend of mine who was studying social science, we look into the number of positive cases of corona per 100,000 population at a time when the country went into lockdown. So you can see that there is a very nice line of best fit that separates the countries where those countries that went into lockdown quite early when they had quite few cases, compared to some countries that went into lockdown when they had quite a high number of cases. So it's not surprising, like for example, in the United Kingdom where I'm currently stuck in, Cancer Research UK issued a statement on how the pandemic has or is going to affect cancer management. So, so the Cancer Research UK projected that about 200,000 people per week are now no longer being screened for bowel, breast, and cervical cancer across the UK. And there is certainly a drop in the number of urgent referrals for cancer. So what this means is that, or what this could mean is that patients with symptoms of cancer or probable cancer are less likely to come forward, potentially with because they have the fear of contracting the virus in the hospital setting. And this is going to have a domino effect. In essence, it's going to lead to delayed diagnosis, delayed treatment, and potentially an unimaginable impact on outcome and survival. And so I think as a cancer researcher, I think we ought to understand what are the wider implications of COVID on in our research setting. So of course, no one can do research alone. I certainly can't do it by myself. So with me throughout my journey, I need to thank each and every one of these individuals for certainly making my research experience a memorable one. It wasn't always easy, but I guess if you have the right people along the way, it certainly makes the journey more worthwhile. So I thank you for your attention, and I'm very happy to answer any questions that you may have.
Right. Thanks a lot, Sean. Yeah. Wow, that's a very good presentation. At least I'm learning more actually about your research in your masters. Well, I have one question here. I think it's a burning question by Apple.
Question.
How many papers have you actually published? Okay.
I normally I normally answer that question myself at the end of the year.
Okay.
But I guess I would say it's about 10, I suppose.
10 papers.
Yeah.
Wow, that's good.
10-ish, I think. Yeah.
So that's like throughout your journey in research.
Yeah.
Okay.
Yeah.
Wow. Okay, that's good. Congratulations. Right. Okay. Here's a burning question that I guess most people would ask a cancer researcher or someone who's doing a research on cancer, or someone from a cancer background. Is diet important in helping to control cancer? For example, by avoiding sugars or carbohydrates.
Yes, I think that's certainly an unavoidable question.
Yeah.
I mean, I literally see it coming because it's the kind of questions that cancer researchers get all the time.
I know, right? Yeah.
And okay, so I'm going to do my best to answer, but don't shoot me if I'm not going to give a direct one. So if we're talking about controlling cancer in terms of managing risk, then my answer would be, I think yes, because we know that obesity is certainly a risk factor for developing cancer. And I think Malaysia is the fattest nation in Southeast Asia, right?
Yeah.
I saw it somewhere. So
Too much good food.
yes, if you want to lose weight, you come to the UK. I guarantee you lose weight. So so yes, so I would say in terms of controlling cancer risk, there's certainly a link between cancer and obesity, and also cancer development. In terms of controlling cancer when you have cancer, I think that's probably a more difficult question for me to answer, mainly because I think at the end of the day, it's about taking a whole-rounded approach, talking to your doctor about the other risks that you can manage or other things you can do to manage your cancer. So I would think that a nutritionist or a dietitian might be in a better place to answer that question.
Okay, all right, thanks. It's a very general answer, but I guess people will get the idea of what we are saying. Like, yeah, you can't really look at cancer researchers and ask this question because we don't have all the answers actually. All right, I'll go to the next question. This is a question from Tom. So, is blood cancer generally more deadly than other cancers because it's already spreading in the bloodstream?
Okay, so when we talk about survival and cancer, it's always a very tricky question, because each cancer would have its own different subtypes as well, right? So I guess very famous examples are breast cancer, for example. So breast cancer is not just one disease; it's actually made up of multiple disease. And one way to stratify breast cancer is that you look into the kind of receptors, hormonal receptors or growth factor receptors that are expressed on the tumor cells. And typically, patients that express the hormonal receptor in breast cancer tends to do better in terms of survival compared to cancer patients whose tumor don't express any of those receptors because there's no target, isn't it, right? So
Yeah.
even within cancers, you can already see some heterogeneity in terms of aggressiveness and therefore survival. And the same thing goes for blood cancer. I think blood cancer is even a more heterogeneous disease where you have many, many subtypes depending on the cell of origin, and more importantly, the kind of genetic mutation. So there are certain genetic mutations that confer a worse survival, typically because those patients have more aggressive disease. And there are some patients who get sort of like a favorable genetic mutation that who turns out to survive or to outdo their other patients. But in terms of aggressiveness, coming from the point of view where it's releasing immature cells, I would say patients typically, perhaps they don't so much pass on because of the spreading of cancer in blood cancer patients, but they typically pass on due to infections or bleeding. Because in for example, acute myeloid leukemia, the stem cells are defective and therefore they release a lot of immature cells into the bloodstream that can no longer do its normal function, such as fight off infections or for blood clotting.
Okay. Yeah. Yeah. All right. So we'll move on to the next question. But before that, the picture that you put, the rice, the one on the rice.
Yeah.
Yeah, that's from Uncle Nigel, right?
Yeah, that was my.
Yeah. The chat box was going crazy when they saw that. Wow, I tell you, you have to look at the chat box. Like everyone was like, "Oh my God, that's the colander and the rice."
Yes, I do keep myself updated with memes as well. So that's one way not to cook your rice, people. I can
Yes.
give a separate tutorial on that if you need help.
Okay. All right. Here's a question by me. Okay. I saw the one where you said that you were studying like single cells, right? Okay. So do you think the interaction with the microenvironment of the tumor may have actually been neglected in the analysis when you are actually analyzing only single cells? Because the microenvironment of the tumor is also playing a big part to how they survive and whatnot. So do you think maybe that data may have been neglected when you only study single cells?
Not at all. I think when
Okay.
we do look into single cells, we are going to inadvertently capture some of the microenvironments as well. And so what we do is that we just do the sequencing on all the single cells. And then after that, downstream, you try to use some gene signatures to separate the tumor cells, and then you separate the microenvironment cells, and you separate the immune cells as well. So mind you, the immune cells are also part of the microenvironment, and you will definitely capture it in the single cell approaches.
Okay, all right, okay, understand. Here's a question by Hakim. So he's asking, this is when you were saying that you only did the dry lab, whereas the people in the lab, the lab scientists are doing for you the wet lab. So he's asking, how do you work as a team with the wet lab scientists?
Yes, so I guess that's the beauty of it, isn't it, right? Because I definitely can't do my work if it weren't for the people in the wet lab. So a big shout out to them for taking the time to optimize the protocols, because it takes time. It takes heck a lot of
Yeah.
time. So so how do we work as a team? That's exactly how we work in a team, because everybody have their own expertise that they bring to the team, and so everybody is quite good in what they do. So we therefore can move the project quite quickly. So the key to move quickly from there is really about effective communication, because you're going to speak to someone with a potentially different expertise. But once you get past that, I think the project moves very, very quickly.
So these lab scientists, are they actually like they are hired people in the lab, or are they students as well? So do you add them into your published work? I mean your papers?
Well, at the end of the day, I think the position in which someone holds in the lab, whether you're a student or just or whether you are a postdoc or research assistant or very senior person, we close one eye on position, because we're only interested in what kind of expertise you have in a team. And as long as you contributed your expertise to the project, I would say the right thing to do is to have them acknowledged.
All right, here's another question by Hakim. Can you determine the stage of cancer via gene expression?
Ah, I think there are some studies out there that certainly try to derive gene signatures that are associated with more aggressive subtypes. So yeah, I'm sure there are works going on out there to sort of to extract this kind of gene signatures. And I think a lot of these studies are certainly done post hoc. So they probably have some indication on which cancers are aggressive or and so on and so forth, and then they do a comparative analysis, and normally that's also validated in a prospective cohort potentially. But yes, there are studies out there that are looking into this.
Oh, I'm sorry, I think the question just now was from Dr. Izzat, but it's okay. Sorry, Dr. Izzat. All right, let's move on to the next question. The next question is, does your research include new cancer treatment?
That is the goal. That is the goal. We are trying
Okay.
to we are certainly trying to look for biomarkers or genetic mutations that can respond to existing therapy. That would be nice because you know how long it takes to make a drug, isn't it? It takes 10 to 20 years to make a drug. That's like somebody who has begun his postdoc and then after that is preparing for retirement at the age of 50 plus. So it takes forever to make a new drug. So, but that's one way to go about, you can look for a new biomarker and then from there develop a new drug because sometimes the effort is worth it. But I think my philosophy is that I want to identify new biomarkers and see whether those biomarkers are responsive to existing FDA-approved drugs. Because I think the pipeline for repurposing existing drugs that has been approved will be much faster compared to developing a drug from scratch. But it depends on
true.
yeah. So I'm not saying that one method is better than the other. I'm saying that you should just leverage on your expertise and go with that.
Yeah, totally agree with you. I think we should look at because producing a new drug is not easy and it's a long journey. You have to go through preclinical, clinical, and whatnot. Oh my God, too much of things.
Yes, life is too short. Sorry.
Yes. Yeah, life is too short.
Again.
From either patient xenografts, organoids, I did imagine the number of cells or cell population is very different. Do these samples reflect or complement each other well in single cell sequencing experiments?
Right. Okay. So I guess I failed to mention where I got the samples from or where the lab got the samples from, right? So essentially, we get it straight from the patient.
Okay.
We get it straight from their bone marrows or their whole blood, and so we directly perform the sequencing reactions on those samples. We don't culture them to organoids or cell cultures before we do the sequencing, although there are some people doing that, but that's not what I'm looking into. And I think just to dovetail from that question, I think that's the kind of research that I want to continue to be in, because I think the real answer lies in the clinical samples, and I want to continue to look for teams that actually go straight to the heart of the problem and look and try to understand cancers.
Yeah, true. Looking at the clinical samples will be much better, and also I think we can directly address the problem instead of going through animal study and whatnot, right?
Yeah, but animal studies and individual studies are still very important. For example, for drug testing, isn't it? Because somehow you just have to culture the cells out of the body and then test your high throughput drug systems, right? So I guess it's really not about trying to find which method is better, but it's about trying to find complementary methods to get answers.
Right. Okay, here's another question. So, following the question about the new treatment ways. Okay, this is a follow-up question. So, which place would provide better facilities for your research? Is Malaysia good enough to help you in fulfilling your goal?
I guess that's a lot of questions that people, I mean, that's the kind of question that we ask ourselves, right? Towards the end of graduation, what hope at the end of the tunnel? How to answer this question? I think it's always about playing to your strengths, isn't it? Right. So if you're like me right now and you're overseas and you're kind of like studying patient materials and all that, I think right now, of course, being in working in the Western side of the world, the focus has very much been on Caucasian patients, isn't it, right? And
Yes.
there's always a niche wherever you go, as long as you can think about that. So if I were to one day come back to Malaysia and try to meet my goals, then I think there's still a niche for myself and other aspiring Malaysians who wish to return to Malaysia if they're overseas. So Asians make up more than half of the world population, isn't it? But
Very true.
more than half of the research are looking into Caucasian patients.
Caucasian.
I'm
Yeah.
not saying that the findings in Caucasians are not applicable to Asians, but there are
Mhm.
some settings where it's more suitable to study in an Asian population, such as, for example, it was just that week that I learned that nasopharyngeal cancers are more common in Asians,
Mhm.
and also what's that cancer?
Colorectal cancer is also common in East Asian.
Sorry, which one?
Colorectal cancer.
Um, I'm not too sure about that.
Yeah. I'm telling you that colorectal cancer is also more common among East Asians. So yeah, you can't really look at data from the Caucasian side too much.
I mean, you can, but
Yeah.
maybe the etiology may be different as well.
Mm-hmm.
It's possible. I'm guessing.
Mm-hmm.
I think bile duct cancers are also more common in Asians, but even so, potentially, our genetic makeups are different, isn't it? Right? And sometimes
Yep.
our genetic makeups may have an impact on cancer treatment and all that. So I think right now, I think all these are question marks, and I think we can certainly answer that kind of questions if we return to the East. So there's always a niche wherever you go. All right. Yeah.
Okay. Wow. There's so many questions now. Be prepared. Okay. Another question by Haki from single cell studies. Would it be possible to create a more effective cocktail for cancer treatment?
Yes. So right now, I mean, the world began. The world began 50 years ago. I think chemotherapy was first tried out in blood cancers in children, isn't it, right? And then so chemotherapy and radiotherapy really became the mainstay for cancer treatment. And I think right now in the 21st century, the world is moving into a more targeted therapy approach. So we have already seen that, for example, in certain breast cancers that express the HER2 gene. So these are targeted specifically using the Herceptin drug, right? And then for breast cancers that express, for example, estrogen receptor on its surface, that specifically target using certain inhibitors that are very specific to those protein. And then blood cancers, I think in a subset of blood cancers in which has the a kind of fusion genes, that also has been, there's a drug to specifically target it. And so that began the single agent approach or single drug approach. And what I can see from the literature is that people are slowly moving to combine targeted therapy. So using more than one drug, like 2 or 3, to target patients. Because remember, the tumor itself is not just a homogeneous ball of mass. It consists of many different subpopulations that may have different kinds of mutations. So yes, I think a single cell approach can delineate the different kind of genetic mutations, and you can therefore correspondingly, or at least hypothetically, pick and choose the drugs that correspond to each of the mutations.
Okay. All right. Yeah. So we actually have quite a few more questions here, but we don't really have much time. I'll just go through a few of them, and then we'll see whether we can finish it up.
Okay.
Yeah. So Sabrina has a question where what aspects do you find most challenging working with cancer single cell data?
Okay. I would say it's the heterogeneity of it, because I never expected the cancer, even if you look into a single patient himself, you never expect the tumors to consist of so many smaller components. And it's about trying to delineate or distinguish the different cell populations that was especially challenging. And more importantly, trying to understand the biological difference between this different cell population. And finally, what does that mean for cancer diagnosis and treatments? So I would say it's not just the wet lab part where the protocols are challenging, but also the bioinformatic part is also, I think, equally challenging as well.
I think Sabrina is asking that because I guess she is also a bioinformatician, so she could relate to you. Yeah. All right. So we'll move on to the next question. Is questions from Sunket. He actually has 4 questions, but I guess we don't have the time for that. We'll try to address 2 of it. Okay. Sunket from Facebook. So he's asking how many single nucleotide polymorphisms or mutations of BRCA1 or 2 genes are known to be associated with breast cancer so far.
How many? Okay.
Yeah.
Okay, so I guess I need to break down I really need to break down that question into 2 parts, right? So if you get in terms of because the gene is, let's say, this long, correct? Let's say it's arbitrary this long. The reality is that the mutation can happen anywhere along the length of the gene, correct? So theoretically, there can be really a lot of ways in which a gene can be mutated along the gene length, and that can confer an increased risk to get cancer. So typically those mutations are like, small deletions or maybe small insertions, or maybe a nucleotide change, for example. But sometimes that in some populations, it is almost always that one particular same mutation that you find, in the Jewish population. And I think it's just that 3 mutations that is commonly found in like more than 90% in those breast cancer patients that are Jewish, right? And then the second part of that question is single nucleotide polymorphisms. So single nucleotide polymorphism is very, very tricky because I don't think there's one single nucleotide polymorphism that increases your risk dramatically to get cancer, but you have to look at it in the context of a polygenic risk scoring. So I'm not going to go too much in depth into it, but a polygenic risk scoring just means that you don't just look into one polymorphism, but you look into multiple polymorphism, and then you assess that person's risk based on that collection of polymorphism.
Okay. Wow. I mean, man, you're really, really knowledgeable in this, man. Okay, so let's move on to the non-science part of it. Okay. There's 3 questions. Okay, we'll end it with a non-science questions.
Yeah.
Right. Hakim is asking, is Oxford as stressful as some people say?
I wonder who told you that. At the end of the day, when we ask questions about a person's experience, I can tell you that every person's experience is going to be unique. The least that I can do to answer that question is to list down some factors that can certainly influence your life. It's not just in Oxford or studies, but it's also applicable to work. So I think hopefully you get to do a project that you are genuinely interested in, and genuinely believe that that's going to change people's life in the future. That's one motivation. The second is also about having very good chemistry with your colleagues and your bosses. That's also going to play a role. And the third is probably the weather. Do you like the 4 seasons? Do you hate winter, or are you a sunny person like me? So what I'm trying to say is that everybody's experience is going to be different. So yeah, I say live in the moment.
Okay. All right. And what motivates you to dedicate most of your time in this work field? Is there any story that you would like to share?
Most of the time. Oh, I didn't admit to that. But anyways, yes, I guess, I think there are 3 factors to cancer, isn't it, right? There are 3 ways, 3 main risk factors for cancer. The first is whether you inherited a genetic mutation or not. If you do, then maybe you may have an earlier onset. The second is environmental factors, right? These are things that you can control, like smoking, okay? That goes without saying for lung cancer. And then there's a third factor that sometimes we don't really talk about, which is age. Because as you grow older, your cells will have gone through many, many rounds of replication already. And that, sometimes if you're unlucky, it's going to accumulate a lot of mutation. And if you're even more unlucky, it reaches a threshold where it becomes tumor. So in a sense, I think I hope to genuinely believe that I'm making a difference, or I'm going to make a difference to patients. And that's why I'm focusing so much on therapy, because I'm asking the question, okay, you have cancer now now what? Can we do more for you? And at the same time, I think at a more personal level, it's about really facing my own mortality.
Okay, right. And one final question. What are your career aspirations after your PhD? What do you plan to do?
What do I plan to do? I don't know. I guess it's like this year, right? Nobody expected Corona to happen, isn't it? So I guess...
Carpe diem, live in the moment.
It's carpe diem. At the same time, at the back of your mind, you should also be planning. But I guess I can't tell you specifically what I want to do, is because I don't know myself. But I guess broadly what I want to do is that I want to do I want to be involved in meaningful work, work that has an impact and change the way that we do things for the better. And once I look at that definition, I no longer confine myself to just cancer research, but hopefully I can still be involved in cancer research because I want to be part of a community to solve this very complex problem and to make a difference in cancer patients' life. So yes, that's where I hope to move on.
All right. So yeah, thank you very much, Sean, for sharing with us your research. I'm really mind blown at the amount of papers you have published and the amount of work you have done and your experience. So I also like to thank everyone who have joined us today. All right. So next Saturday we will be joined by Shazeli Zain, YSN ASM Chrysalis Award finalist, and he will be sharing with us how an underutilized agriculture waste material can be transformed into a high quality product. All right. And for more updates regarding the future Research Insider series, like our Facebook page, Embryos and 100 Scientists of Malaysia. So thank you everyone and see you next Saturday. Thanks, Sean.
All right. Bye-bye.
Bye.
Bye.